Hello students, today we're going to be starting chapter 10, which is all about DNA and how DNA works and what its structure looks like and how they discovered it. So to start with today, we're going to be looking at DNA structure and how it was found, or how they discovered that DNA was the genetic information. You can see here kind of what the DNA will look like. We have some things we're going to be covering today and the next day: adenine, guanine, cytosine, and thymine, and those are in here. You can see that that kind of makes up part of the DNA. The DNA is called a double helix, and it's this twisted ladder. Inside of here, we have one of these cytosines, or Cs, is made up of a sugar, a phosphate, and a nitrogen base. So we'll be going through all those things here in the next day of notes. So first, the discovery of DNA. We have Griffith's experiment, which was in 1928. And his experiment showed that there's hereditary material that will pass from one bacterial cell to another. So pretty simple, but just saying there is hereditary material. It is passing from one cell to another. And when that happens, we call that transformation. So this word here you can put in there. If you're using the notes packet, you can see there's a section for Griffith's experiment. And transformation just says we're transferring genetic material from one cell to another. And that's really all you have to put in. You don't have to do the added part there with one organism to another. We would assume that if it's going cell to cell, that that's also happening within a larger organism. So basically how he did this experiment, this is on page 194, but he took these mice, he injected a mouse with live R cells. So R, it actually stands for something, but you can just have the abbreviation there. If you're using the notes packet, you have this picture in your notes packet. So the R cells do not kill the mouse by themselves. He injects the mouse with live S cells, a different type of bacteria, and the S cells do kill the mouse. So then we want to see, okay, now we know that R cells keep the mouse alive, S cells kill the mouse. We want to see if we take the S cells and we kill them, so the cells are no longer functioning, with heat, which is something like you would do if you're cooking something. You kill off any bacteria or something that might be there with heat. That's why we would cook food. And he injects this mouse with the heat-killed S cells. He finds that the heat-killed S cells cannot kill the mouse. So then his experiment 4 is he takes the S cells with the heat, he mixes them with live R cells. So at this point, we're looking at the S cells which can kill the mouse. They're dead at this point, but they're mixed in with the live R cells. And he injects the mouse with the mixture. At this point, we see that the hereditary information transfers from the S cells, so that dangerous S cells, it transfers to the R cells. And so the cell itself was dead, but not the hereditary information. And so that hereditary information basically took over that live R cell and then caused that mouse to die. So this was the point of transformation, or transferring DNA from one cell to another. So our next scientist here, Avery, in the 1940s, and he showed that DNA was hereditary material that transfers information between the bacterial cells. So this isn't too much different than what was already discovered, but the key part here is that he called it DNA, or discovered that it was the DNA that was being passed. So our next scientist, Hershey and Chase. These two worked together to determine that DNA was the actual hereditary information. So they are credited for confirming this, and credited when we look at hereditary information and understanding DNA, even though there were scientists before them that led to their experiment. So they confirmed that DNA is the hereditary material. And the way they did that, there was a thought at the time that it was either protein or DNA. And so the way they did this was take a virus, and a virus is only made up of either protein or DNA, that stuff that's inside. Now these viruses look a little bit like little spiders, and what they'll do is inject the material into a living cell, and then that DNA will take over that cell and will then continue to replicate this viral DNA, which then is what will make an organism sick. So what they ended up using was a bacteriophage, and you have this on your notes packet, this picture, but it's not drawn in really well. So you may want to draw in or add to the picture that you have. And a bacteriophage basically is a virus that will kill or eat up the bacteria. And so a virus is made up of only a protein covering and then the DNA inside. And so they set up two experiments. One was that they took the protein covering and they labeled it, protein covering, labeled with radioactive sulfur. They then allowed it to inject a bacterial cell. And so you can see there to infect this bacterial cell. And what they found was that there was no radioactive material in the cell. So this would help to eliminate protein as the hereditary information. So their second part of the experiment was to take this DNA and label it radioactive phosphorus. So in this experiment, they did the same setup. They had it infect a bacterial cell, and what they found was the radioactive DNA in the bacteria. So this is when they concluded that DNA is the genetic information. Now, you have this picture on your notes packet, and you will probably want to go ahead and write some extra things down to make sure that you have the understanding of what the picture is showing you, because it didn't copy very good if you're using that notes packet. And basically, the results here, the results showed DNA was being passed from the virus to the bacteria. Another thing you can note is that it is on page 195 if you want to go there and look up some more pictures. All right, so moving on to the structure of DNA. And again, this is Chapter 10, Section 2. We're going to combine these two sections in class as well. So in this one, we're going to look at our actual structure. We're going to be looking at our nucleotide. We'll look at how these nucleotide bases are strung together. And then also this outer part here, the double helix ladder. Okay, so first, the DNA is the blueprint of life. The two scientists that were credited with coming up with this picture or image like this was Watson and Crick, and you'll see those in your book as well. It was in the 1950s, so really not all that long ago considering how much we know about DNA now. DNA does contain the instructions for making proteins within the cell, and this is kind of a key part. We're going to get to this more in Chapter 10, Section 3, as far as how the DNA makes up the proteins. The shape is a double helix, and it's made up of repeating subunits called nucleotides. And so we'll look at that shape kind of what a nucleotide is. You can see here in the drawing we have thymine and adenine and guanine and cytosine. We'll get into those on another slide here, but you can see they're connected, and notice they're in repeating patterns. On the outside here we have our sugars and our phosphates in the blue and the green, and then there's hydrogen bonds that connect these nitrogen bases. We'll get into all that on the next couple slides. So first you have a place in your notes to put in a definition for a gene. DNA codes for genes, and then genes are segments of DNA that code for a protein. So genes code for a specific protein. The protein then in turn codes for a trait. Those traits, some examples are skin tone, eye color. Here you have a gene for hair color, you have a gene for tooth development, a gene for a specific protein, maybe an enzyme. And to kind of show you how all this fits together, we have nucleus here, DNA is inside the nucleus, showing you kind of what that DNA looks like if we were to string it all out. And then these little spots here are codes for a specific gene, which then in turn code for a specific trait. All right, so deoxyribonucleic acid, DNA, stands for deoxyribonucleic acid. You can see there where the DNA comes from. The monomer for DNA is nucleotide. This, just as a reminder, we did this in chapter 3, so it kind of gives you a little focus back on what we've covered before. Each nucleotide is made up of 3 things: the sugar, in this case deoxyribose sugar, a phosphate group, and then one of 4 different nitrogen bases. These 2 things here make up the sides of the ladder, and this portion here makes up the rungs of the ladder. Now, you have a picture in your notes packet, and if you're not using the notes packet, you need to draw this picture in of what a nucleotide is. This is a nucleotide. And if you have the notes packet, it didn't copy very well, so let's make sure that we put those in. You aren't showing in your notes packet the phosphate group. Make sure you label that. The sugar. And then we have the nitrogen base, so it's 3 parts. Make sure you have that labeled correctly. Okay, the 4 bases, so this portion here, there's 4 choices: thymine, cytosine, guanine, and adenine. You can represent them with just their letters, T, C, G, and A. To get into them just a little bit more, G and A, guanine and adenine, are called purines, and the key about them is that they have a double ring of carbon-nitrogen. And then C and T are called pyrimidines, and they have a single carbon-nitrogen ring. So basically what it will look like is kind of a puzzle piece as far as how their carbon and nitrogen ring or bonding looks. In this, you can see here what that means. So adenine, double ring of carbon-nitrogen. Guanine, double ring of carbon-nitrogen. Thymine and cytosine, single rings of carbon-nitrogen. Okay, so complementary base pairing, C and G are always matched together, and T and A are always matched together. You can see some examples here. T and A bonded together, C and G bonded together. Now these bonds right here are called hydrogen bonds. And A and T are always put together with 2 hydrogen bonds. And C and G are going to be put together with 3 hydrogen bonds. You have a place on your notes packet to put that. Show you on the next slide. So how are these bases put together? Hydrogen bonds, we have 3 hydrogen bonds, C and G, 2 hydrogen bonds, T and A. If you have your notes packet, you can just fill those numbers in. The rest of the sentence is given to you. Okay, so that's our basic structure of DNA. Go back to a picture here. The basic structure of DNA. This right here is our sugar and our phosphate, again making up the sides of the ladder. And then these here are your bases that make up the rung of the ladder. And then it would be twisted into a double helix.